Imagine a world where we can do computation inside living cells.
The problem we're trying to solve is really trying to have a more sophisticated diagnosis that can happen automatically inside cells.
Imagine a biological computer that operates inside a living cell. So, for example, it could be used to determine whether a cell is cancerous, and if so, then trigger the death of the cell.
So here we're talking about little molecular systems that you can, work that run in a test tube or maybe even in a live cell, so they're really small. The type of work that they're doing is essentially they're trying to sense, analyze, and control molecular information.
In this project, we're trying to use DNA as a programmable material.
DNA is highly programmable, just like a computer, and we can program a whole range of complex behaviors using DNA molecules.
So we're taking advantage of a phenomenon called DNA strand displacement.
So DNA strand displacement is essentially a competitive hybridization reaction. So it's two strands of DNA that come together, and as they bind to one another, a third strand that was initially bound is kicked off. One issue with any biology research or biomolecular research is that it's always sort of a cycle of trial and error. You test it, it doesn't work, so you go back to the drawing board, and you do that over and over again. And it's a slow cycle because doing experiments just is hard and it takes a lot of time.
We developed a language for programming molecular circuits made of DNA. So the programmer would write down a collection of DNA strands, and the software will work out how these DNA strands interact with each other and can be used to predict their behavior over time. And this kind of software could, for example, be used to detect and fix bugs in a molecular circuit design before that circuit is built. For decades, biologists have been using chemical reaction networks as a means of describing the behavior of biological systems. So what our technology enables for the first time is that a system described as a chemical reaction network can now be translated and implemented in biology at the molecular level.
One of the things that we've done recently, which I'm particularly excited about, is that we have created, designed, using our tool, and created experimentally is an implementation of the so-called approximate majority algorithm. At the moment, really, the technology is very much in its infancy. It's still very much at the research stage. So most of what we're doing is in the test tube. An enormous goal will be to have what we're able to do in the test tube also working inside cells. That's a hugely enormous challenge.
So this could enable a whole range of biotechnology applications. For example, it could allow us to both detect and treat disease to a level of precision that has not been possible so far. It can also allow us to make new compounds far more efficiently. These compounds could be medicines or biomaterials, and ultimately it could allow us to make biological computers that operate at the molecular scale.
So I started my professional life about 30 years ago as a nurse in the pediatric intensive care unit. And I remember this one infant, let's call him Jonathan, who came in really, really ill. Seemed to have a rare genetic defect, but in those days, gene diagnosis was still in its infancy, so we couldn't really figure out what's wrong with him. And in the years since, as I've trained as a physician scientist, we've been living this phenomenal digital and scientific revolution. And I'm here today to tell you that we're actually hacking the software of life, and that it's changing the way we think about prevention and treatment of disease. So here's all the biology you need to know in 30 seconds. Our body is made out of organs. Our organs are made out of cells. And in every cell, there's this thing called messenger RNA, or mRNA for short, that transmits the critical information from the DNA, our genes, to the protein, which is really the stuff we're all made out of. This is the critical information that determines what the cell will actually do. And so we think of it like an operating system. And it's not just in every cell of our body, it's actually in every cell of every organism alive. It's the same thing. And so if you could actually change that, which we call the software of life, if you could introduce a line of code or change a line of code, it turns out that has profound implications for everything from the flu to cancer.
True. Peace. Kens, I'm going to hit this article up. It's called Software of Life, man. Keep the slang, right? Software of Life, Evidence for God's Existence. Man, check it out. The software of life, the blueprint for assembling the protein part for cells and organs in correct timing and order, are encoded into our DNA. DNA is digital code, similar to binary computer code, although it is quaternary, having 4 letters instead of 2. The density of the information encoded into DNA staggers the imagination. There is enough information stored in space in a half teaspoon of DNA to store all of the assembly instructions for every creature ever made, and room left over to include every book ever written. The information content of a bacterium has been estimated to be around 10 billions of whatever bits of information, comparable to 100 million pages of Encyclopedia Britannica. Even if the insurmountable problems with the structure of life's hardware assembled, mentioned in the last chapter, were somehow abridged, another completely separate phenomenon is needed for this life to operate and reproduce: a DNA language. Man, peep this slang. Software of life, DNA code. The information stored in the software of life, DNA, is encoded by way of various arrangements of 4 molecules called nucleotides: adenine, guanine, cytosine, and thymine. Okay, y'all get it. These molecules are arranged in chains, bonded together into what is called a double helix. A double helix, which looks like a ladder that has been twisted into a corkscrew shape. Just as with binary computer code, this seemingly simple structure can store vast amounts of information for the assembly of proteins throughout the organism. If all the DNA info in the one human body were printed in books, it would be enough to fill the Grand Canyon 50 times over. Moreover, the information will be nothing but gibberish and will be worthless for constructing proteins unless there was an established language convention to which it conformed. This raises the question: who established the language convention? Man, peep the slang. As mind-boggling as this seems so far, it gets even better. The instructions for creating the DNA double helix are encoded into proteins. This raises the question: which came first, the DNA or the proteins? We have seen clear evidence in the hardware of life that neither proteins nor DNA could be expected to arise spontaneously, but here we see the problem multiplied exponentially. If life arose by random chance, we could have to have 1 amino acids assembling spontaneously in the correct order with the correct chirality, all left-handed molecules to create 2 proteins, 3 nucleic acids assembling spontaneously to create DNA simultaneously with the arousal of proteins with the correct, I'm gonna have to look up this word, chirality, right-handedness in the correct order, the following the same very specific complex language convention in order to orchestrate the assembly of proteins. One is tempted to ask the question: what came first, the chicken, proteins, or the egg, DNA? But in this case, both phenomena would have to arise simultaneously and following the same language convention, or they will both be completely useless. Man, peace to slang. Software of life, the riddle of the genetic code. When examining the software of life, Nobel Prize winning biochemist Jacques Monod said the major problem is the origin of the genetic code and of its translation mechanism. Indeed, instead of a problem, it ought rather to be called a riddle. The code is meaningless unless translated. The modern cell's translating machinery consists of at least 50 macromolecule components, which are themselves coded in DNA. The code cannot be translated otherwise than by products of translation. It is the modern expression of omni vivum ex ovo. Everything that lives comes from an egg. When and how did this circle become closed? it is exceedingly difficult to imagine. Monod is not the only Nobel Prize winning scientist to cast doubt on a naturalistic explanation for life. Consider the worlds of Lila Progogine, okay, chemist, physicist, and recipient of 2 Nobel Prizes in chemistry. The statistical probability that organic structures and the most precisely harmonized reactions that typify living organisms will be generated by accident is 0. Now keep in mind that the conditions mentioned above are necessary but are not sufficient for the creation of life. There are many, many more challenges for the theory of spontaneous generation of life that we don't have room to discuss here. Suffice it to say that it takes more faith to believe this could happen by chance than to believe in a transcendent creator who has orchestrated it. Software of life, powerful argument for design. Peter Slang, modern science. This one I want to get to, continues to delve into the software of life, consistently revealing new, mind-boggling features of the cellular world on a regular basis. The nanotechnology that we observe functioning as this incredibly tiny level is overshadowed only by the incredible complexity of the language and encoding which orchestrated and is built into it. Man, I done went back on some. Peep the Slang, let me finish. Arguably the most prominent atheist of the 20th century, and Tony Flew, professor of philosophy, author, and debater, announced in 2004 that he had become convinced that there is no way these things could have evolved by chance due to their stunning information content. As he put it, it now seems to me that the findings of more than 50 years of DNA research have provided materials for a new, enormously powerful argument to design. Man, I done went back on some of my old videos, and I tell you, they ain't good at all. Man, Peep the Slang, yo.